Hey there, welcome to lesson 7.12. This lesson continues to focus on herbivorous or plant parasitic nematodes, describing root lesion nematodes in the genus Pratylenchus. So let's jump into lesson 7.12. So let's look at Pratylenchus, the root lesion nematode. It has a very wide variety of hosts, that's over 400 plant species that feed Pratylenchus, and that includes cereals, coffee, corn, bananas, and other fruit, legumes, and potatoes. And you can see here in the image this stylet that Pratylenchus uses to get into different plants. There are over 68 species of Pratylenchus worldwide, and it is the third nematode in rank in terms of the most severe economic impact that plant parasitic nematodes have. It is migratory in nature, and it is an endoparasite. That means it lives inside the plant root. It creates lesions by feeding on the plant, and the lesions are a form of necrotic discolored patches on the root. And you can see some of these patches here, and some of the damage that is caused in a scanning electron micrograph. And here are the nematodes traveling and living inside the plant root. And in this particular case, this is a capsicum, and Pratylenchus capsici is the species of Pratylenchus in this particular case. There are at least 8 species of Pratylenchus that affect cereals. Pratylenchus neglectus and thorny are found in over 90% of dryland wheat in Pacific Northwest USA, and in Australia, where cereals are grown in the northern regions, and that's in Queensland and New South Wales. Both thorny and neglectus have been noticed since 1960s, and in late 1970s, thorny was demonstrated to cause yield losses in wheat. Around $33 million worth of damage is attributed to Pratylenchus thorny annually in these regions, and elsewhere overall up to 69% of yield losses are attributed to Pratylenchus thorny and 32% to Pratylenchus neglectus. Here in the image we can see a wheat root on the left that has numerous tillers and lateral roots, as opposed to a root that is heavily infested with Pratylenchus neglectus, showing lack of root development as a result of this infection. Pratylenchus has various penetration mechanisms depending on the host species that it feeds on. It is attracted to the sites of root hair production and root tips. For example, Pratylenchus penetrans rubs its lips along the epidermal cells, but doesn't penetrate the cell walls of the cells. In lucerne and in clover, it penetrates the main roots at a site where the lateral roots rupture the cortex and migrate via the main roots cortex into lateral roots. In turf grasses, they aggregate and penetrate at the zone of root elongation, while Pratylenchus thorny penetrates chickpea roots without site preferences. So there are just various ways, depending on the host plant and probably the conditions within which the plant is growing, the specific conditions within the field, as to the various mechanisms the nematode uses to penetrate into the plant tissue. Here is a beautiful image showing Pratylenchus inside the root tissue of a plant. So let's look at the Pratylenchus life cycle, and we'll begin here at a point where the nematode penetrates the root and then invades the root cortex. The invaded tissue then turns brown, causing lesion, and the nematode reproduces and migrates within the root, and this results in a reduced root system and lesion development on the roots of the infected plant, and some of the nematodes will leave at this point and infect other roots. The invaded cortical tissue will collapse and break down due to the infestation, and the nematodes will either leave the decaying roots and infect new roots because their food supply by then has run out and the plant struggles to provide the necessary nutrients as its root system is affected, especially when the young roots are killed. And after the nematodes have reproduced, their eggs are laid in the plant and end up in the soil as the plant disintegrates or are directly released into the soil. In the egg, the first stage juvenile will develop and undergo a first molt, and the second stage juveniles will leave the egg and undergo a second molt, resulting in the third stage juvenile and a third molt. And then the last juvenile stage is stage 4, and this molt results in the adults being formed, which are able to breed. And all of the stages from the second stage to the adult stage are able to infect roots. And when they say here healthy plants, this really is questionable because it is mostly plants that are weakened that allow a heavy infestation by nematodes. Healthy plants might have a few nematodes feed on them, but it will never result in such terrible damage to the plant where it results in its death. So just to go a little bit deeper into this life cycle of Pratylenchus, both adults and all the juveniles except for stage 1, which is in the egg, can penetrate, migrate and feed within the roots. Pratylenchus thorni and neglectus are parthenogenic. That means that the females can produce fertile eggs without the need of being fertilized by a male. Pratylenchus penetrans is amphimictic, which means that it does require sexual reproduction and fertilization of the eggs by a male nematode. The J1, or the stage 1, develops within the egg, which we've already mentioned, and molts to the second stage juvenile and hatches. And then the J3 molts into J4, and then the adult is then a reproductive stage of the nematode. So all the stages increase their size and sexual development, and the life cycle can occur completely within roots or on root surface or in soil. The life cycle typically lasts between 4 to 8 weeks, and that depends on the species of Pratylenchus, the food abundance and type, as well as temperature, the conditions of the host, and the moisture that is available. Pratylenchus can undergo 3 to 6 generations in one growing season, and eggs will either be laid in roots or directly in the soil, but when the root disintegrates, the eggs will end up in soil. The eggs provide a long-term survival in adverse conditions, so they are very hardy and make Pratylenchus very difficult to control. And this is another example of a life cycle that I think is beneficial to go through once more, as it has additional notes. So let's go through it. So the nematode invades the outer layer of the root cortex, and it enters the stele of the plant, of the root, via the cortex, so to the inner core of the root tissue. And this feeding causes the cortical tissue to turn brown and collapse and break down. The highest number of nematodes occurs within the 25 centimeters of soil, but they can be found much deeper in some soils, and that depends on the depth that the roots penetrate into the soil. The eggs are laid in the roots, and nematodes reproduce and migrate within the roots and soil. And also some eggs are laid directly in soil as the nematodes travel in between plants. So here are the different stages of development, as we have already mentioned. And the nematodes survive summer in soils within root pieces or in a desiccated state, which is important as it allows their survival. So they're very resilient, and there may be several generations of nematodes each growing season. As described earlier, when it rains, the nematodes become rehydrated and once again become active, so they can invade the growing plant roots of the next crops or any weeds that are in the field, or any other plants that are available to them. And because of their very varied diet, their love of various plant species, it is easy for them to maintain their populations within the field, unless specific measures are taken to reduce their numbers. So the symptoms, as already described, are normally lesions that lead to degradation and sloughing off of root epidermal and cortical cells. And this reduces root branching and therefore water and nutrient absorption by the plant, and leads to the degradation of lateral roots and loss of root hairs. Lesions rapidly coalesce, and this leads to this extensive discoloration. The plants end up being stunted with premature leaf yellowing and with reduced tillering and weight, and therefore reduced yields. The lesions favor root rotting and saprophytic fungi and bacteria, allowing these organisms to feed on and decompose the affected plants. And symptoms are often confused with nutrient deficiencies, drought, and root diseases, such as fungal diseases in particular. And cortical degradation and reduced root branching is often confused with Pythium rhizoctonia root rot. So oftentimes this is misdiagnosed. You can see here in the image on the right how in the carrot, the nematode actually produces prolific lateral roots, and this is Pratylenchus penetrans. So the carrot doesn't really develop, so instead of developing its normal carrot growth, it will produce this lateral root proliferation. So let's now look at how Pratylenchus is controlled in the field and what are the methods of control in order to prevent economic damage to crops that is associated with Pratylenchus. Well, obviously, when crops are grown in monoculture, that increases the population density of the nematode and yield losses because the entire paddock is filled with food source for the nematode, and basically there's not much effort the nematode has to do in order to invade the plants because it just moves from plant to plant. So growing crops in monoculture is definitely the number one problem. And this is why in natural habitats where there is species diversity, there isn't much problem with root lesion nematodes or any other parasitic plant nematodes because it's all balanced and the plants are able to develop resistance, and there are just not as many plants for them to feed on, or they will move on to other plants, obviously, to feed on as well because they love to eat many different species. Which is good because in nature that means that one species is not eliminated. But when we grow things in monoculture, there is just this very severe infestation, and oftentimes, and mostly because those plants are already under stress. So crop rotation is difficult because of the polyphagy within the nematode, so the ability of the nematode to feed on various host plants. And there is very minimal information on crop rotation in wheat in particular. However, there are some examples where Pratylenchus thorny has been reduced significantly by rotation of wheat with corn, cotton, or soybeans for 2 consecutive years, and this was shown in Sonora in Mexico. A barley cultivar called Clipper in Queensland in Australia has been shown to reduce Pratylenchus thorny as well, while Pratylenchus Z, which infects rice, has been shown to be reduced when legumes such as mung beans and black gram are rotated with the rice. So it is very essential to know exactly what you're doing when you're trying to use rotation as a method of controlling Pratylenchus. Here is a beautiful image of Pratylenchus eggs in root tissue. Removing weeds, ensuring sanitation in the field is another method of control to prevent the survival of the nematode. Unfortunately, also, this is kind of a way of reducing biodiversity in the field, so they will always then feed on crops. But you can also grow antagonistic crops, such as marigold. So there are some plants that Pratylenchus doesn't like, and once again, the knowledge of these particular plants is necessary, and they can be used after cropping or used between crop rows to control the nematode. Ploughing is often used, and this is, of course, damaging to all the soil creatures. So while Pratylenchus will be basically chopped up by the ploughing mechanism, that's the whole role of ploughing, is to homogenize the soil, and a lot of the times it just kills the organisms. But this will also result in the killing of the nematodes that actually feed on Pratylenchus, such as predatory nematodes, which actually prefer to feed on plant parasitic nematodes. Another way to control Pratylenchus is through solarization with polyethylene film, and this has been demonstrated in chickpea fields and has to happen for about 6 to 8 weeks to have a reduction of Pratylenchus thorni by 50%. And that's once again a very detrimental way of control to the soil ecosystem, because you're heating up and exterminating all the beneficial organisms as well that cannot withstand high temperatures. And so this is basically sterilization, you could call it as well. Other ways are to use seed dressing or coating or soaking with carbosulfan in certain crops. And this is very toxic to the entire ecosystem, including humans that are going to consume the crop, the seeds, as chemicals often get into, and there are residues within our food after such treatment. And then carbofuran can also be applied as well during sowing. Once again, toxic way, an expensive way to control the nematode. It has been found that nitrogen fertilized barley has a greater abundance of root lesion nematodes over a 22-year period versus non-fertilized plots. So there's obviously some interaction going on where the plants are more susceptible to infestation when they are fed synthetic nitrogen fertilizer. And this beautiful image shows an adult female of Pratylenchus penetrans. So the most economical and ecological way of controlling root lesion nematodes is by using resistant and tolerant cultivars. So resistant cultivars actually reduce reproduction of the nematode and therefore reduce yield losses, while tolerant cultivars grow and yield well while under nematode pressure. So the nematodes still proliferate, but the yields are unaffected or not as affected as they would be in susceptible cultivars. So tolerant cultivars don't decrease numbers of the nematodes and allow their reproduction, but will increase yields by up to 30%. So the best method is to combine resistance with tolerance and grow varieties that have both these capacities. The image on the left shows Pratylenchus thorni intolerant variety and its distinct yellowing and reduced vigor despite good rainfall, as opposed to tolerant variety here on the right, which is obviously more vigorous and greener. And of course, it is a lot of work to find these varieties, and a lot of money is invested in creating cultivars that are resistant and finding those that are tolerant. And both of these have to be developed. So there is a lot of work involved in that. And we will discuss the importance of soil health in general and the microbial diversity in the soil ecosystem as to being the best way to actually control plant parasitic nematodes, because these organisms actually have natural predators, and in particular predatory nematodes that love to consume the sweet plant parasitic nematodes. Because remember, these plant parasites specifically feed on the sugars in the plant, so they are very attractive to other creatures that love to have an easy snack on them because they're high in energy content straight from the plant. So predators naturally are attracted to these organisms. At the same time, if we plow and use chemicals and do all these nasty things to control the nematodes, the predatory nematodes will also be killed off and just keep increasing the problem in the soil, making the soil and the plants more susceptible to plant parasitic nematodes. To demonstrate how soil health and certain agricultural practices affect Pratylenchus thorni, the Queensland Department of Agriculture on the Grains Research and Development Corporation website has a study that describes a field trial at the Hermitage Research Station in Warwick, Queensland, Australia. And what they have done was tested various organic matter amendments to the soil of between 0, obviously a control, to 20 tonnes per hectare, which was actually incorporated into the topsoil. There were also 3 cropping treatments, so continuous fallow, so basically testing whether having no crops present in the soil would reduce Pratylenchus. Then there was sorghum crop grown as well, but no residues of that crop were retained. And then there was a sorghum crop grown, but the residues were retained. With the increased organic matter treatment, as well as the sorghum treatment where the stubble was retained, there was an increase in beneficial nematodes and a decrease in root lesion nematode numbers. And the trials have found that there was an increase in soil biology in the continuous cropping systems versus when the fields were left without crops in fallow, that the continuous cropping actually had a greater effect against Pratylenchus as opposed to the addition of up to 20 tons of organic matter per hectare. They've also found that the soil chemical and physical differences, as well as nitrogen and phosphorus applications, had no influence on the soil's suppressiveness of Pratylenchus. So this is an image showing stubble retention and what it looks like when crop residues are not removed, but that they are retained in the field. So it is known that intensive machinery and inputs actually increase soil compaction, and this decreases soil porosity and therefore root development. So as opposed to that, an increased organic matter content is actually good for the soil. It decreases compaction and increases soil porosity and therefore root development, and as a result it reduces the numbers of nematodes. And that's because the plants actually have a greater root development and are healthier and therefore less susceptible to being consumed and attacked by nematodes. Organic matter increase in the soil also increases cation exchange capacity and therefore plant nutrition, and once again that helps the plant to defend itself and interact on a more healthy level with any nematodes that want to consume the plant. The trial has found that no tillage, stubble retention, and diverse organic matter inputs increase microbial activity and soil structure, and therefore increased suppressiveness of Pratylenchus in the soil. The presence of continuous crop in the field means that the plants continuously feed microbial diversity, and also the heavy stubble retention was shown to increase microbial activity and therefore suppression of Pratylenchus in the soil. And I'd like to emphasize that these three components, the organic matter retention or addition, no-till, as well as stubble retention practices, actually encourage the soil food web to proliferate. So this diagram basically represents a simplified structure of the soil food web just to demonstrate how the different components of the soil ecosystem relate to one another and why soils are more suppressive to parasitic nematodes when the whole ecosystem is healthy and all the players of the soil ecosystem are present. So as we have already learned in previous lessons, plant parasitic and other nematodes are consumed by parasitic fungi, by nematode-trapping fungi, predatory nematodes, as well as mites. So obviously plant parasitic nematodes serve as a food source for these creatures. And also there's nutrient cycling through the various nematode types, as well as predators of bacteria and fungi, which include protozoa and some of the smaller invertebrates. And that causes nutrient cycling to occur and allows the plant to access those nutrients, as well as having these microbes present on the roots. And what is missing here is the mycorrhizal fungi that actually protect their plant host and oftentimes compete with plant parasitic nematodes. And of course, the plant in itself exudes a lot of food for all of these organisms, but particularly feeding the bacteria and fungi on which the rest of the organisms feed. And the organic matter that is present is also food for bacteria and fungi, and therefore everything functions as it should as long as there are healthy plants and lots of organic matter in the soil as the primary food sources for the soil food web to function. After testing 130 soils, the trial found various numbers across those samples of the various organisms that cause suppressiveness of soils to root lesion nematodes. So, for example, nematode-trapping fungi were found in 42% of the soils, predatory nematodes in 77% of the soils, parasitic bacteria, in particular Pasteria, in 25% of the soils, and root colonizing fungi were also present. And all of these increased resistance of plants to the root lesion nematode. Glasshouse experiments showed that when soil was sterilized and then when 10% of suppressive soil was added to sterilized soil, it decreased Pratylenchus numbers at the rate of 60 to 90%. So when the soil is sterilized and there's no biological activity present and the parasite is added to the plants, there is a decimation of the plant because there are no other organisms in the soil to suppress the parasitic feeding by Pratylenchus. And so this means that suppressiveness is in fact biological, but also transferable. So adding some of the members of a healthy soil ecosystem into the soil can have a positive effect on suppressing Pratylenchus in sterile soils. Suppression was also greater at between 0 to 15 centimeters in the soil compared with soil lower down the profile. And this makes sense because this is where most of the biological activity occurs, within the topmost 10 centimeters of the soil surface. And this image is just a reminder of some of the fungi that we've already spoken about, the nematode trapping fungi that consume nematodes and therefore reduce their numbers in soils, protecting plants. And it's noteworthy that plant parasitic nematodes actually are pretty sweet. So they have a lot of carbohydrates, a lot of carbon in them that the fungi love to consume. So they probably really do appreciate feeding on plant parasitic nematodes as a direct carbohydrate source coming straight from the plant. But the overall principle here is that there is a need to improve the soil ecosystem in terms of biodiversity of organisms in order to reduce plant parasitic nematodes, and in this case we've just spoken about Pratylenchus, or the root lesion nematode. So this is the end of lesson 7.12, and in our next lesson we'll look at the role of herbivorous nematodes in natural ecosystems, the benefits of these creatures. So they have a role besides just eating our crops, and that's something that we'll look at in lesson 7.13. So you'll be hearing from me again then. Bye for now.